Hello, this is Thomas Warwick, welcoming you to this Bite-Sized Bio webinar, which today is sponsored by Twist Bioscience. Today's presentation is titled The Power of CRISPRgate for Transcriptome Engineering, Innovations in Screening, and is being presented by Dr. Philipp Rathert from the University of Stuttgart. Philipp Rathert leads a group of scientists investigating disease-relevant molecular epigenetic mechanisms. In particular, the research focuses on the understanding of chromatin networks in different cancer types. He is an expert in the development and application of functional genetic tools such as RNAi and CRISPR-Cas9 in multiplex screening approaches, and he has long-standing expertise in protein biochemical and cell biological methods to understand the underlying epigenetic process in more detail. He has industry and academic experience and has been an independent research group leader in the Department of Biochemistry at the University of Stuttgart since October 2015. As always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears at the top right of your screen, and I'll put them to Philip at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Philip, for the presentation.
Yeah, thank you for the nice introduction. I hope you can see my screen and the pointer. And I was asked today to present to you our new addition to the CRISPR toolbox, which we termed CRISPRgate for gene and transcriptome engineering. And I will give you a brief introduction into the technology and how we came up with it. So first of all, a quick recap. So when I started my studies, basically, loss of function methods were reduced to one main basically tool that was available, which was RNAi, which is or was at that time and is still a great tool to quickly induce the loss of function of a certain knockout. And it basically targets the transcript, so therefore it's reversible. However, you have competitive inhibition, a huge variance in knockdown efficiency, so you have to very carefully design your sgRNAs or siRNAs to achieve a beautiful or effective knockout. And in addition to that, we have other problems like sequence independent and dependent off-target effects, high or low expression of the RNAi influences the knockdown. And that obviously is not optimal in different screening setups, so it can skew up your results, and it's difficult to compare different RNAis. And when I was in my postdoc at the IMP in Vienna, basically this loss of function toolbox was extended by the identification of CRISPR at that time, where Jennifer Doudna and Emmanuel Carpentier also achieved the Nobel Prize in the past years for that. And basically, you all know that CRISPR, short for clustered regularly interspaced short palindromic repeats, is basically a defense system of bacteria against invading foreign DNA, where this foreign DNA, parts of this foreign DNA are integrated into the CRISPR locus. These parts of DNA are processed, and then during this processing are forming the sgRNAs that are then bound by Cas9, and these are complementary to the foreign DNA again, so that the bacteria can defend against viruses, for example. Mechanistically, it looks like this. So we have the Cas9 protein that is also encoded in the CRISPR locus. And once the Cas9 together with the sgRNA has bound the DNA target site, and the sgRNA is matching to the DNA target site and identifies the PAM sequence, the H&H domain and RAFC domain use this energy that is formed during the formation of H bonds here to perform a conformational change and bringing the the cleavage sites very closely to the DNA, to the target DNA, and then this target DNA is cleaved by the HNH domain and RAF-C domain. Why I'm going into this detail will become important in the talk later. So basically what is then, or how the CRISPR technology was then later developed in, was that the tracer and the sgRNA were fused together to make the design a little bit easier. And what the tool was then used for is basically produce genomic knockouts. Well, the Cas9 binds the sgRNA. This is designed to a specific locus, and in this locus, it cleaves the DNA. And due to non-homologous end joining, this DNA locus is repaired. Basically, what happens then, we have error-prone end joining here. So we have a premature, or we can achieve a premature stop codon due to erroneous repairs. We can have frame shifts, and this then in the end leads to a truncated or a skewed up protein, which then induces a loss of function. But the technology was then further developed into a kind of reversible tool, which is not inducing knockouts on a genomic level, but it produces knockdowns. For this, it was mutated, so the Cas9 protein was mutated, so the cleavage sites do not function anymore, and it was fused to a functional domain. And in most cases, or the most potent one that was identified in the past years, is the CREP domain of ZIM3. And then basically sgRNAs are designed to target not the exons anymore, but the promoter region. And in this promoter region, after binding the CREP domain, then induces a relatively strong silencing and a reduction of mRNA. And in the past years, these technologies are used in various settings, and I'm just highlighting the most easy and the most straightforward ones here. Basically, what is performed are the loss of function CRISPR screens, basically, where people look for gene essentiality in certain cell lines or cell types. Basically, we have cell lines that express the Cas9 protein. sgRNAs are designed to target different genes transduced in the cell line, and then after cultivation, and the cultivation varies from 7 days to 30 to 40 days, and then the dropouts in this screen are identified as essential genes. And we have done something like this in the past. And other screens, for example, look for drug resistance, basically same setup, but at a certain point, the cell culture dishes are split into a control group, and the other one is treated with a drug. And if you have synergistic effects, for example, the knockout of a gene induces sensitivity against a certain drug, you have more dropout cells and can compare this then to the controls. This is also something that we have done here in the past. And what becomes more and more fashionable is that you look for biological effects. And there you use, for example, readouts like EMT, where you basically look for genes that induce a certain biological effect. And by inducing a loss of function of these genes in a screening approach, you can screen for genes that are, for example, essential for transition from epithelial to mesenchymal stem cells, like in a screening-based approach, or use reporter genes that can report on the function of a certain gene, which we have also done in the past and published. However, what we have realized in all these screening ventures and looking at screening approaches of other labs is that these screening outputs are not always black and white. And one big problem is if you, for example, look at CRISPR knockout screening approaches. Ideally, you produce a premature stop codon or a frame shift where the protein at a certain point is then degraded and not functional anymore. However, what comes very quickly in recent publications is that you obviously not always have an erroneous non-homologous end joining repair here, but you also have in-frame repair, which leads to a perfectly fine protein in the end and doesn't give you any loss of function effect, or you produce or the cells start to produce alternative transcripts of your protein, which is a big problem. And this is, for example, showcased here in this publication where the authors looked at, I think it's 50 to 100 different proteins after CRISPR knockout. And they quickly realized here, for example, in red, that the mRNA of most of these proteins is still present, which is not a big surprise, but also is the protein amount. So this was then quantified by mass spectrometry, and they realized that obviously a few proteins are depleted to a certain extent, but many are still expressed at a relatively high level, which means we have in-frame repair or alternative splicing leading to still functional protein products. And this obviously will mess up your screening approaches. On the other hand, also the CRISPRi approach is not completely or 100% effective. So for example, we can have genes which have very strong epigenetic regulation or are highly transcribed. So the polymerase and co-regulators of the polymerase keep these loci open, and you will have a hard time in producing effective knockdowns of certain genes. And on the other hand, we have genes with multiple TSS transcription start sites where you realize that you can design sgRNAs for a non-functional transcriptional start site, and the protein is still produced from an alternative one. And in addition, it is very important where you position the sgRNAs at the transcription or around the transcription start site, because this is shown here. Not all sgRNAs designed for a certain promoter will also lead to a very strong knockdown. So these problems have led to sgRNAs or sgRNA libraries where people tended to use more sgRNAs per target gene. They started to focus on sgRNAs with very high efficiency, so high knockdown efficiency. And there are a lot of examples where people try to optimize sgRNA predictions. However, the increased number of sgRNAs and this higher this focus on high efficiency also increased off targets, and all these points then led to large sgRNA libraries with significant heterogeneity in the final output targeting, for example, the same gene. And this is obviously a problem for the next generation of CRISPR screens that we are looking forward to, basically where we try to screen single cell or perform single cell RNA sequencing combined with certain dropout of genes, or in vivo screens in organoids, or even primary patient samples. Because here it's critical that your libraries are relatively small, because the number of cells that are available here in all these setups are very limited, or it's very expensive to sequence deeper. And this is, for example, showcased for an example here where people performed an in vivo screen using a primary patient cell samples. And the authors of this paper here, for example, extracted patient samples from PDAC patients. They transduced these with 40 sgRNAs because they targeted 4,000 genes with approximately 10 sgRNAs per gene. They selected them and then transduced them in a mouse. And in this scheme here, it looks really pretty straightforward because here they transduce this in one mouse, state that this is in triplicate. But if you look into the methods, you realize they aimed for 600,000 x sgRNA representation, which means ideally they would inject these mice with 24 million sgRNA positive cells, which means this is quite difficult. And actually 8 million cells per mouse were injected, which would be 200x representation. So they needed 3 mice per replicate. So 3 mice, 9 mice in total for the experiment, which is relatively little. If you think about you have a huge mouse facility, but in the end, if we want to reduce animal burden and animal testing in our experiments, this is still quite a lot. So basically, we thought about something that we could do in terms of improving the consistency between our sgRNAs targeting the same genes, and thereby reducing the size of the sgRNA libraries. And there we came up with the idea that we could combine CRISPRi with CRISPR knockout. Basically, keep the Cas9 intact, so keep an active Cas9 enzyme, and combine this with the CREB domain of ZIM3. And through the provision of 2 sgRNAs per cell, we could then target Cas9 to 2 positions, basically one to the promoter region to induce a knockdown of the gene transcript, and on the other hand, target the active Cas9 to a common exon of the same gene and induce a DNA cleavage. However, this is difficult because normally after cleavage and repair, the sequence here of the genomic DNA is obstructed, and this means Cas9 would not bind this sequence anymore. And in this region here, in the promoter region, we need basically the presence of our construct or of our fusion protein all the time. So this has to be stable. And there I'm coming back to the conformational change this enzyme is doing after it has found its target. Because this is a paper from 2017 where people could show that basically the conformational change is dependent on the exact pairing of Cas9, basically. And this you can see here in this experiment where basically this is a FRET experiment where this domain was fused with a FRET probe, and the Cas9 protein itself has the acceptor probe, and then once the conformational shift was done, the authors could observe a change in signal. And once you achieved, or once they introduced more than 4 base pairs mismatch, they could see that this conformational change is not happening anymore, although the protein is still bound to the DNA. And this was something that we thought we could use to make the initial binding here at the promoter region stable, because we didn't need an active Cas9 enzyme, but we just needed binding there. So, and this was then our final setup. In the end, we had an active Cas9 fused to a Sim3 CRAB domain. And then we provide the cells with a dual sgRNA construct where we have, on the one hand, a 15 nucleotide sgRNA, which targets basically Cas9 to the promoter region. And there, Cas9 was supposed to be inactive, but Sim3 could still induce silencing of the promoter. Additionally, we have a 20 nucleotide sgRNA, just standard sgRNA targeted to a common exon. And there, Cas9 would still be active, cleave the exon, and induce a genomic mutation or an indel, basically, so a knockout of the gene. So obviously, we had to validate our idea. And the first was that we wanted to check if indeed our truncated sgRNAs recruit Cas9 to the target region. For this, we used a reporter construct that was stably transduced in a reporter cell line, where basically we recruit dead Cas9 fused to Sim3 CRAB to a promoter with TET-O binding sites, and we designed the sgRNA to bind the TET-O binding sites and just truncated it from the 5' end. And what we observed is that basically we could truncate until 14 base pairs, basically, and achieve the same reduction in expression of our reporter fluorophore. So that was settled. So we can still recruit Cas9 with 15 nucleotide sgRNAs. However, does it still cleave? This we wanted to determine in a different experiment. So we had two basically reporter genes, CD13 and CD3. These are surface proteins on leukemia cell lines, for example. And we targeted those genes basically, first of all, with a 20 nucleotide sgRNA. And there you can see that we can induce a very nice knockout of CD13 and CD33 if we use a 20 nucleotide sgRNA. However, this was different with a 15 nucleotide sgRNA. Here you see that the Cas9 is not active anymore, and it doesn't induce any knockout of our target genes. Only, and this was our first hint that our technology could work, and basically we selected the perfect sgRNA predicted, or the perfectly predicted sgRNA for CD33. And this was unfortunately very close to the transcription start site of this gene. And the SimCRAB was still fused to the protein. So when we see that, when we functionalize the active Cas9 with Sim3, we didn't get cleavage, but it was so close to the TSS that it induced silencing. And this is basically what told us that in theory, the technology could work. We quantified the indels then by deep sequencing and indeed saw that the 15 nucleotide sgRNA does not induce cleavage, whereas with the 20 nucleotide sgRNA, we see a lot of indels occurring at CD13 and CD33. So basically, yes, we can use this technology. However, we were initially a little bit worried that due to the truncation of our sgRNA by 5 nucleotides, we would obviously increase the number of off-targets because, well, the sgRNAs are shorter. And then obviously Cas9 could bind. at additional genomic regions. So what we basically did is we used again that Cas9 fused to the CREP domain, and then performed RNA-seq after targeting Cas9 to CD33 with 50 nucleotide sgRNAs and the corresponding 20 nucleotide sgRNAs. And here we specifically selected sgRNAs that have a lot of additional perfect off targets in close proximity to a TSS of a certain gene. Because we wanted to push our system or to push Cas9 to bind to additional genes and induce a transcription downregulation. So we did RNA-seq, and we can see that controls and wild type clustered very nicely together. sgRNAs, so once we looked at the transcriptome alterations after transducing the sgRNAs, we saw that indeed we see knockdown of genes. But to our surprise, and this you see very nicely here, the the 20 nucleotide sgRNA and the 50 nucleotide sgRNAs, the corresponding ones, clustered very tightly together. And this was something that we basically didn't expect. And you can see that a little bit better here. If we compare sgRNA1 and sgRNA2, the 15 mer and the 20 mer, we see that basically the transcriptional alterations induced by both are very similar. So there is not a big difference. And if we do this a bit close or look at this a little bit closer, we see obviously there are a few changes between 15 and 20 mers, but they are very mild. And to our surprise, they are not affected by mismatches. So most of the genes that were deregulated basically were not targets of our sgRNAs. And this we see here for the second sgRNA as well. And if we look at all genes, we see that basically there's no big difference between the 15 nucleotide and the 20 nucleotide sgRNA. And that was very surprising to us. And we looked at a few genes a little bit closer. Basically, they are already highlighted here. But if we look at these genes here, for example, we see that they are mildly downregulated in all cases. But this is pretty much independent on the number of mismatches, which suggests basically that Cas9 can still bind to loci even if it contains a huge number of mismatches, even here, for example, for EGR1 in the seed region. However, since our system does not produce more off-targets than, for example, CRISPRi, we've still continued working with this, and then decided now to look into the improvement that we could achieve in more detail. So again, we used a leukemia cell line looking at our surface markers, CD13 and CD33. We can very nicely image those or monitor the expression of these proteins using targeted antibodies, and then analyze the knockdown efficiency by flow cytometry. So if we transduce our construct with sgRNAs, so the CRISPRgate construct with sgRNAs targeting these surface markers, basically we can monitor the loss of these surface markers over time using flow cytometry. And as you can see here, if we look at CRISPR knockout or CRISPRi, we see that basically they can induce a change in the expression. But if we combine both sgRNAs in our CRISPRgate construct, we can improve this effect. Similarly, if we have a strong CRISPR knockout and a weak CRISPRi, also this combination is improved here. And if we have two mediocre sgRNAs in CRISPRi and CRISPR knockout, the combination will still greatly improve both. And this you can see here basically over time, where you see that sometimes the difference is not that high, but in some cases it's really significant. So we tested a huge number of different sgRNAs, and basically this is now a variety of sgRNAs with different efficiencies predicted. And you can see that CRISPR knockout and CRISPRi over time, they achieve a certain knockout, but if you look at the variance between those, it is really, really huge. So what happens if we combine this now? There you can see that we get similar variance in the beginning. However, we are already relatively significant. And in the end, all our sgRNAs, so basically these sgRNAs are combinations of those here. And we can see that we basically rescue most of the mediocre sgRNAs that lead to the large variance here in our CRISPR-Gate setup, which told us that indeed, using this approach, we can induce or we can basically reduce the variance in CRISPR screens. So that means combining CRISPRi and CRISPR knockout improves the screening setup. So the next was we wanted to look at genes that are hard to target, because I told you initially that some genes, even if you target them with sgRNAs and try to produce knockouts, it's basically impossible. They get repaired either in frame or splice variants are expressed. And then there's one example which we found in the literature, which is BAB1, nothing that we normally would study, because this is a protein that was long debated and to be very important during mitosis. So during the segregation of the chromosomes, it plays an important role. And it was very debated initially because, well, initially it was non-essential, which is a little bit surprising if you think about its effect in mitosis. Then different papers showed that it's impossible to produce CRISPR knockouts only if you design two sgRNAs that cut out the complete gene. It was possible to achieve a knockout of BAB1, which is not surprising if you look at this gene. It has a lot of different splice variants and a variety of different transcription start sites. So we wanted to know if CRISPRgate could achieve basically what this group has achieved by a full knockout. And for this, we selected the top 3 predicted sgRNAs for CRISPRi and CRISPR knockout and combined those, always the top 1 predicted sgRNA for CRISPRi with a top 1 predicted sgRNA for CRISPR knockout in our CRISPRgate system, and compared this effect then to CRISPRi and CRISPR knockout alone. And there you basically see, again, we see this large variance between the top 3 predicted sgRNAs in CRISPRi and CRISPR knockout. However, in CRISPRgate, this is not visible. We achieve always a very strong and a very small difference in between the different sgRNAs. And we also compared this then to the gold standard of basic loss of function screens like CRISPR-off or or the benchmark that was published here in this paper. And yes, the benchmark performs similarly well. CRISPR-off not so much. However, we could still improve, or we can achieve the same effect as the benchmark in the end. And we also looked then at cell cycle deregulation, and we see that with CRISPRgate, we achieve a lot of, or we can achieve alterations in cell cycle comparable to the benchmark, sometimes even better, and and to CRISPR off. So that told us, well, yes, we can improve actual loss of function studies using this approach. However, we also wanted to know, can we use this technology in relatively sensitive cells? And one cell type that was always or is always very sensitive to DNA damage, which is basically something that we induce with our system, is embryonic stem cells. So we looked at a human iPSC cell line, which has a stable integration of basically two genes, neurogenin 1 and neurogenin 2. So upon the addition of doxycycline, these two genes get expressed. And if you then remove also a few growth factors, these iPSC line develops, differentiates basically into neurons. So what we wanted to test now in this experiment is, can we interfere with the process of the differentiation by targeting the transgene? So in this case, we would work against a very strong promoter, basically. And on the other hand, we wanted to know, if we target a non-essential gene like ART1, for example, can these cells still develop into a neuron? So this is what we basically did. Here you can see these cells before the addition of doxycycline and after the addition of doxycycline, and you can see that at a certain point, after 3 days, these have developed into neurons, and we see the upregulation of a neuronal marker. So when we then transduce them with our CRISPRgate construct and different sgRNAs targeting ART1, so the non-essential gene, or neurogenin 2, basically you can see that before that, the iPSC marker protein TRA160 is expressed. If we induce then neurogenin expression and we have no ZIM3-Cas9 expressed, they downregulate this embryonic stem cell marker. And this is also the case if we express Cas9, ZIM3-Cas9 with the ART1 sgRNA, but we can interfere with the differentiation process if we target neurogenin 2, which told us basically that indeed our system induces DNA damage, but it is still possible to be used in sensitive cells. So the next thing that we then wanted to do is, and here we teamed up with Twist Bioscience, we wanted to perform a screen, basically. So what we did is we designed a focused sgRNA library where we target 100 or over 1,100 genes, all associated with chromatin. So our lab is working in the field of epigenetics. So this is basically what interests us. So we target chromatin-associated genes with 3 genes per gene. 3 sgRNAs per gene. And then we transduce our cells, we select them, and then cultivate them for basically 14 days, which is 7 passages. And in the end, we extract the sgRNAs from genomic DNA, we amplify this, send it for sequencing, and then we calculate the difference between day 14 and T0 to identify those sgRNAs that were targeting genes that are essential for these cells to proliferate. And this is basically a very standard dropout screen. However, it's just performed for 14 days, which is relatively short, but we have seen in the previous data that the CRISPRgate effect is very fast. So since we wanted to be fast and don't spend much time in complex and difficult cloning strategies, we talked to Twist about the synthesis of something that is a little bit bigger, because in our constructs, obviously, we need to pair a 15 nucleotide sgRNA for a certain gene with corresponding 20 nucleotide sgRNA for the same gene. And in between, we need a tracer and a promoter sequence. And this obviously makes our construct relatively large. There are cloning strategies in the literature and out there where you basically can synthesize the 15 nucleotide sgRNA and the 20 nucleotide sgRNA with a cloning site in between, and then clone this into the vector of interest. But as I said, we wanted to be fast and we didn't want to spend a lot of time in optimizing and implementing these in our lab. So we talked to Twist if this synthesis of 270 base pairs would be possible. And they said they are working on something like this and gave us access to this platform early on. So we were able to get these large oligos from Twist and cloned them into our backbone. And as I was told at that time, this was basically the largest or the longest oligo that was synthesized in a pool synthesis performed by Twist at that time. We were very excited. The initial chimera rate was very low. So basically, the chimera you could achieve with different. Well, the chimera is something that we basically don't want. So there you combine one 50 nucleotide sgRNA with the wrong 20 nucleotide sgRNA, which could happen during the PCR through template swapping of the polymerase, or due to homologous recombination in the packaging cells or E. coli, for example. However, the chimera rate was exceptionally low, below 10%. During our processing, that was marginally increased, so also something that we could work with. And in the end, we had a result that looked very, very promising. So we had a couple of internal controls here, positive controls, so controls where we knew the cells are sensitive against. In red, they cluster here and deplete over time. Neutral controls don't. And if we then look at essential genes or non-essential genes, you see that the sgRNAs are very nicely clustering here on the left side, whereas the non-essential genes are non-depleted over time. So this gave us hope that our screening approach worked and the design was good. However, we needed something to compare our screen to published screens. And initially we thought about, yeah, let's look at the log fold change. But this is difficult because the log fold change always depends on, first of all, the cell doubling time and the cultivation duration. So at the passages that were performed after these cells were transduced. And interestingly, if we looked then into the literature, these passaging times, even if the same cell line was used for the screening approach, passaging times were varying massively. And if you, for example, compare a screen like ours with 7 passages with a screen that has been passaged 30 times, this is a big difference in the log fold change. And so there, since we wanted to screen only a short period, we couldn't compare the log fold change. So what we came up then as a measure was the delta log fold change. So look at the variance between the sgRNAs that target the same gene. And this difference here we call the delta log fold change. And in addition, we decided to look at the p-value of the genes in the screening setup. So we selected then a variety of different screens that were out there where people have either screened leukemia cell lines or optimized screening libraries. And as I said, this was performed in different cell lines, but we looked at those initially as well. And when we looked at the delta log fold change, for example, against essential genes or all genes, we could see that in most cases, the CRISPRgate led to a smaller delta log fold change in comparison to other screening approaches and libraries. So this means we have a higher consistency in the sgRNA performance, which then would lead to better statistics and hit calling as well. And this was obvious when we looked at the p-values that we achieved. And this is here plotted the log 10 adjusted p-value of the sgRNAs and the genes. And there you can see that indeed, in all cases, the log 10 adjusted p-value here in our approach is higher in comparison to others. So, as I mentioned initially, in our preliminary experiments, we could compare directly the effect between CRISPR knockout and CRISPRi and could see that CRISPRgate is improving these. So we wanted to know if we can extract such information also from our screening approach. And there we used the fact that we designed sgRNAs for the genes that had different transcription start sites. So we designed basically the same sgRNAs or sgRNA combinations for the different transcription start sites if these transcription start sites were more than 1,000 base pairs away. And we combined those with the identical 20 nucleotide sgRNA that then targeted a common exon. So using this information, we could then look at different knockouts and compare those with the functional and the non-functional transcription start site sgRNA. And here we can see that in all cases, if we pair the sgRNA for the knockout with a functional transcription start site, we can see that we can improve the knockdown or the suppression of the gene massively. And also here, when we use the knockout targeting the non-functional gene, we can see that we induce an effect. However, when we then combine it with a functional sgRNA here, we can improve that strongly. And this we see with these two genes here exemplified. Exactly. On the other hand, so this shows that the CRISPRi can improve CRISPR knockout. But on the other hand, can we also do this vice versa? So can we look at sgRNAs basically that we have used and compare those to a screen? And here we used sgRNAs from the human CRISPRi version 2 library from the Weizmann lab. And we combined those with basically a 20-mer sgRNA. So we truncated those and combined them with a knockout sgRNA. And indeed, we can see that our CRISPRgate approach can improve the CRISPRis that were used in this screen in both cases. So here also with the non-functional TSS, we see what's the effect of the knockout alone. So also here, the CRISPR knockout can improve the CRISPRi also in the screening setup. And this we can see here when we plot the true discovery rate against the false discovery rate. Also here for the essential genes, you see that indeed we can improve the effects. So that means all in all, it looks like the CRISPRgate combination is more beneficial than a single CRISPR-I or a single CRISPR knockout. But now, in the recent times, it is more and more fashionable to use dual sgRNA setups to induce stronger effects, either by targeting the same sgRNAs or cutting out larger fragments in a screening setup. So we compared our screen then directly to the dual CRISPR-I library that was published recently. And here, the dual CRISPR-I library targets also 2 transcription start sites. And if you look at the sgRNAs that we used here and combined, we can see that in the combination of our CRISPR-I sgRNA with a knockout sgRNA is still better and improving the effect in comparison to the dual CRISPR-I library. So also here, our CRISPRgate combination is more beneficial than the dual CRISPR-I approach. So having now compared our CRISPRgate against different loss of function libraries, we then wanted to know, this is also always nice, but can we find something new, or did we find basically something new? And there we looked at the DepMap portal, so Project Achilles, which is basically providing, or which provided most of the data here in the DepMap portal, where they screened over 2,000 cell lines in a normal dropout screening setup, looking for essentiality of genes. And then they assigned gene dependency scores to these genes. And what we basically did, we used the gene dependency score from the DepMap portal from our TF1 leukemia cell line and compared the effects of CRISPRgate against those. And what we realized was that, yeah, indeed, we basically tend to find similar things, but we also had clustered after k-means clustering, where we see that basically our CRISPRgate approach leads to stronger loss of function effects. But we also find, obviously, a cluster where the DepMap portal information shows stronger effects against those genes. However, when we look at this cluster here and we plot the DepMap dependency score against our CRISPRgate loss of function log fold change, basically we see this cluster here highlighted, and we see that there are a lot of genes that show a stronger depletion in our screen compared to the DepMap dependency score, which were screened with the Havana library. And here we highlighted several genes which we looked at in more detail, and one is KDM1A and the other one is GFI1B. And I would like to highlight that basically KDM1A and GFI1B are both functioning together. So KDM1A is LSD1, a histone demethylase, and this works together with its co-regulator GFI1B to silence enhancers. And this axis here has not been found or identified in screening approaches before. So we then validated these hits, and here you see again how the sgRNAs performed in our screen. We validated those hits again, and here we did not only compare the standard CRISPR-I and the CRISPR knockout. No, we also used the original sgRNAs that were used in the Havana library, and we also combined CRISPR-I with the Havana knockout just to mimic our CRISPRgate approach. And in all cases, we saw that indeed we improved the screening setup in comparison to a single CRISPR-I or CRISPR knockout. And the combination is not, or the combination of the CRISPR-I and the Havana knockout is not always as beneficial as, for example, the CRISPRgate combination. So since we have performed that, and we could also find new things. So the GFI1b KDM1A axis, for example. We can now safely say that CRISPRgate is another option in the CRISPR toolbox, depending on the experimental questions you have. So, for example, if we look at the initial screen that I've shown you before, where they needed basically 3 mice per replicate, we could now reduce this in our approach. Since we don't use 10 sgRNAs anymore, but only 3, we could reduce this to 1 mouse per replicate in theory. And I think this is something that is very important if you think about all these new screening or all these new potential screening questions that could be answered in vivo single cell or organoid or primary patient sample settings. So as I said, I'm not saying that CRISPRgate is perfect. However, it's a very valuable addition to the current CRISPR toolbox, and depending on your experimental question, this is a very important or can be very important. So in the end, I would like to highlight all people involved. So this is basically my group. Yannis Stadager was the PhD student who was spearheading this project. We collaborated with colleagues at the University Stuttgart, Professor Franziska Traube and Professor Stefan Legevi. We worked together with Professor Stephen Johnson from the Robert Bosch Center for Tumor Diseases. And obviously, we worked together with Twist Bioscience here. Julian Jude was instrumental in helping us to set up the screening and the design of the dual sgRNA oligos for the screen. And the work is funded by different funding entities. So one is the Wilhelm Sander Foundation for Cancer Research, the German Cancer Research Foundation, and the DFG in Germany. So these are all helping me to pay all these people. And with this, I think I'm at the end, and I thank you for your attention. And if you have any questions, feel free to ask them now.